I'm not going to present a lot of hardcore data. I'm going to give you sort of an overview of a commercialization strategy that we've developed at SickKids for translational discovery and the movement of basic science data into the clinical realm and around. So I'm the Associate Chief of Surgery or Surgical Services at SickKids. I'm also a physicist. And so when I moved here from the US, one of the goals that I had was to help revamp our commercialization office on our intellectual property office. And so we've taken certain steps to try to facilitate translational work at SickKids, and I'll give you some examples of that, and I'll give you some case studies both from my lab, but also some from pharma as well, and from some of our pharma partners that are presenting here today, or that are present here today. So I have some disclosures. I have current and past funding agreements with various groups, drug discovery groups, including some that are here, although it won't influence anything that I say. So just to wake you up a little bit, I like sort of philosophers and philosophy. So I think this is a really good sort of quote from Thomas Huxley, who is a anthropologist, I guess, biologist, who talked about sort of the tragedy of science and how all of his hypotheses are getting slayed when you go out and search for a fact. Now, he was sort of a proponent of Darwinian action and was one of the main scientists that promoted Darwinian. I think this really applies to drug discovery today, and a lot of what I'll talk about is generic drug discovery, although I will focus on heart failure and cardiac dysfunction as well. For those of you that are more literature-oriented, he was also Aldous Huxley's grandfather. But so let's sort of delve into this. A lot of you have seen translational medicine paradigms that are linear, and they go from T0, T1, T4, and this very nice linear, delineated, deconstructionist approach. I hate that. I think it's garbage. So what I like is more of a circular paradigm, where you have constant feedback between different mechanisms and different groups of people. So for a lot of people in this audience, and a lot of what happens within heart failure, you're going to start with a clinical problem because that's sort of the definition of what we're doing. And you have to move your way around. You have to figure out what the cellular and organ pathology is, and we heard a lot about that in the last session, sort of very specific molecular mechanisms that are happening, and those molecular characterizations of disease. But at some point, if you want to turn that into an actionable item, and a therapy, you have to move on, and you have to find some target refinement, and you have to start developing drugs, and you have to try testing these drugs. And I don't just mean small molecule drugs, I mean regenerative therapies, I mean biologics, all of these things. They all apply to the same sort of paradigms and approval pathways. So how do you do this? How do you make this useful? Well, I would argue linear is not great. Circular's okay, but actually what you need is two circles. You need to go back and forth, and you need the involvement of everybody involved in these pathways to allow for this process to facilitate itself and to improve discovery and getting actual molecules to the patients that are useful. And so why is this such a problem? So your beautiful hypothesis up at the top, you find your target molecule, you say this is the key to improving cardiac function, and sometime around the bottom of the circle, the ugly facts start to hit you that, hey, I just spent a lot of time and a lot of money working on this and it doesn't work. And so what do? What's the problem? Why does that happen? And I think this is part of the issue, is that there's different talents and different sets of people that aren't geographically or even philosophically aligned. They're involved in this circle. And the earlier you involve everybody in this circle, the faster the process will go and the more efficacious the process will go. It doesn't mean that basic science and fundamental science isn't useful, as I'll talk about in a second. It actually facilitates the process greatly. But if you bring in people early and you allow people to communicate, even if they have different philosophies, you can dramatically facilitate the movement around this circle and improve your hit-to-lead ratios. So the only thing that puts you to sleep faster than my talk is reading venture capital reports and market reports. And so that's what I do. I like to read market reports before I go to bed. So this is an interesting one, but it's a depressing one to me, all right? So what this particular market report from the Kaiser Family Foundation, if you're familiar with them, they publish a lot of really good unbiased data. They looked at annual growth in research spending by condition. And they compared 2005 to 2010 and 2010 to 2013. And you can see of particular interest to this audience is a dramatic loss in the annual end-over-end growth of research in circulatory diseases. Now, you expect some fall off here because right around that time, and the reason they picked these date ranges, was the dramatic loss of on-patent drugs. So everybody knew that investment in drug discovery was going to fall because all of these big name drugs were coming off patent. There's going to be a lot less slush money around to invest with. So not surprisingly, you can see all conditions on the far side, it is also down. But none of these are to the degree that circulatory diseases, which includes heart failure, obviously, undertook. So this is echoed again in sort of the progress or the patient progress that happened. This is data from the CDC, who has a really neat little website where you can customize all kinds of maps and stuff. So on the left-hand side, what I've shown is heart failure death rates, or death from heart failure from 2005 to 7 on the left and 14 to 16 on the right. And you can see on a gross level, there's really no change in the colors of the states, the 48 continental states anyway, and a slight shift maybe in the histogram to a slight maybe improvement, but actually an increase in hospitalizations. And just to show how unbiased it is, there's one of my home states, and we're bad. We're one of the few that got worse. So, you know, this fits and it follows investment pathways, right? And the progression around to new therapies. So this is a table I took from a paper that shows some of, you know, a representative, although a limited set of ongoing trials into new drugs that are used for heart failure. And you can see, obviously, a lot of them are involving the Entresto combination at the top. I would argue that a lot of these targets are pretty unimaginative. These are things that we've hit before, with all due respect to the pharma partners here. These are not new things, and so we need to move the bar a little bit. We need to find some things that are maybe different and allow us to really make a difference and a dent in patient outcomes. And an interesting part of this that went along with sort of the last data I showed you was an article that came up in Scientific American recently that actually was specifically talking about one regenerative medicine therapy that's on the market now or coming up to the market, and Entresto, and talked specifically about the pricing of those and how even the pricing outweighed analyst expectations by quite a bit, by over 50%. So this is not what we owe our patients. We owe our patients better than this. We owe them new therapies, we owe them innovative therapies, and we owe them therapies that are affordable, in my estimation. So how do we take advantage of this system? So this is one methodology which is sort of in various forms in the literature and something that we've implemented. This is actually from a paper that was published using AstraZeneca data or concepts, but it's in various formats in the literature, and I think it's a really, really good sort of thing to keep in the back of your mind as you're looking at drug development. So these 5 steps, finding the right target for the right tissue, and I don't just mean the heart, I mean there's different tissues in the heart, right? So the right sub-tissue in the heart at the right safety profile for the right patient and the right commercialization potential. So AstraZeneca calls this the 5 Rs. It's in various other forms of literature, as I said, but it's a really strong circularization of the concepts that are needed to improve drug design. So there's your beautiful hypothesis at the top, and there's your ugly facts at the bottom. And the earlier you connect those two things, the better. And as sort of academic researchers or translational researchers, I think the things we're the worst at are connecting the yellow box and the gray box. So we tend to think of targets, and we find targets in broad ranges of tissue, and we think that we show them in different model systems that may or may not represent our patient population, that may or may not represent even human physiology. But we've convinced ourselves that they're the right thing, and they're the right thing to test it in. And then when we get down to the right patient and we start thinking about, well, where do we actually test this? We don't know, or it doesn't work. And so what we really need to do, in particular in academic medicine and translational medicine, is better connect the yellow box and the gray box, in my estimation. So let me show you, this is again AstraZeneca data actually, who implemented this process, because I think this is an interesting, I'll show you some academic data too, but this is an interesting pharma side to it that may influence your feelings about bringing in different personalities early in the drug discovery process. So the 2005 to 2010 time epoch is before they implemented this sort of 5R scheme. And on the right-hand side is 2016 to 2012 to 16, where afterwards. Now you can see the number of projects that were undertaken at those two times is substantially different. 199 in the pre, 42 to post. And again, that matches the time when the patents were coming off some of the big name and big income drugs. And so, but look at the difference in success and failure rates as a percentage, right? So a better adjudication of the process and earlier discussions that happen in the process dramatically improve the success. And success here is defined as a drug that made it basically to phase 2 clinical trial. So if we look now at the reason that those projects failed, because I think that's probably the most important thing that we need to bring in at an earlier process. In the early phase, we had all kinds of failures for strategy, like where we're targeting, or processes that we're targeting, the inability to generate chemistry, no leads, even some safety things. But after we have a better evaluation of the initial processes, after we're able to better understand the physiology that's happening in the patient and make sure that we're mimicking that physiology in our testing systems, now we all come down to most of the projects being closed for target validation. And again, this doesn't mean terminating things early necessarily. What it means is better evaluation, that that's the right target at the right time for the right disease, and that you're hitting it in an effective fashion. And just to show you that the importance of fundamental science in this process and mechanistic data, if you break this project closure down into two different paradigms, those that had mechanistic data and those that didn't, ones where there was an underlying mechanism known had a much higher success rate. So again, integrating different personalities, different talent types early in the process to allow these people to communicate and allow for some translation of the information into therapeutic discovery. So if we break that down again by different phases, you can see in the early epoch on the left to the later epoch on the right, we've really shifted our reason for failure into efficacy in the later phases, which is where you should be. You shouldn't be failing for target validation and so forth in the later phases. Those are very expensive failures. You're talking about even a phase 1 trial, you're talking about sort of millions, tens of millions, phase 2 even higher. You get higher than that, you're into the hundreds of millions for a failure. So we should be ending earlier for target reasons, not later. And that's basically what their approach did. So sort of tying this together with the last set of talks, but also tying it with Phyllis, who will talk next, how do we sort of implement better pathways into this circularization? How do we bring in different things? So PHC is personal healthcare information. So if you bring in personal healthcare information early in the process, you dramatically improve success rates. So even at the basic science level, even at the fundamental level, the earlier you can bring in patient physiology, specific patient problems, the better your success rates will be. And that's what these basically showed. So let me show you an example of that. So if we can better integrate rich patient data early in the discovery process and stratify even your basic science data, your fundamental data based on these findings, you can dramatically improve what the outcomes will be and improve your hit-to-lead ratios. So there is no, and I would make an argument, there is no heart failure, and I don't think too many people would disagree with that. There's a broad range of what a phenotype of heart failure is. And so one of the projects in my lab, we started with a broad range of pediatric cardiac samples, both normal and abnormal, and I'd make an argument we don't test enough normal in most of these systems. And we enter extensive omic phenotyping and you end up with these, fancy little network graphs and so forth. But then the key at that point was, because you end up with a list of targets from all of these dysfunctional tissues. You say, how do I decide what to do? I just pick something. But that's not what you should be doing. You shouldn't be just picking something. So we went through and did extensive filtering based on clinical data, rich clinical data, from echocardiograms to medications the patient was on. And we started looking at what molecules, what proteins in our case, were deranged in those specific scenarios. And in fact, we found one. We found a specific target, and we published it in 2014. It's called integrin kinase. And so that's the crystal structure shown on the right with the electrostatic surface. And so we were able to identify that target, and that's now progressed to where we're getting ready for sort of phase 0, phase 1 clinical trials based on small molecule inhibitors. So I think, and we would not have found that had we not integrated earlier patient-specific data. So let me extend that a little bit. How do we improve the model system in testing? Because I think this is also a big limitation. We take, for instance, what the gold standard is, and we assume the gold standard is what we should be testing things in. And we say, well, this has always been used for heart failure, this has always been used for ARVC, or this has always been used for HFpEF. But maybe that's not the right answer. Maybe we really need to find better models that recapitulate what the actual patient is experiencing. And I don't necessarily mean tens of thousands or hundreds of thousands of patients. A very small phenotype. So in this way, we need to phone more friends. I see a lot of stuff coming through our intellectual property office where people will sort of say, well, this is what the gold standard literature is, and I just did it. I said, but did you talk to anybody? Did you find anybody who does this now on an investigational level? And the answer is invariably no. So if you're able to bring in people that have expertise in those areas and can model specific phenotypes, you'll have a much bigger success. So for instance, we have 3 for measurement of function of 2- and 3-dimensional cardiac tissue layers, we've sort of invented 3 different systems. The top system is sold or was sold out of a company in San Diego that was recently bought out by Agilent. There's a poster on that at the back. The middle one is a more recent biosensor we created. There's a poster on that at the back as well. And then the bottom one is actually a new system that isn't patented yet, so there's no poster, where we can now predict drug response and categorize protein, proteome, and or drug changes to model it based on what human physiology is and to tell you what it most mimics in terms of drugs that are already on the market, all right? And using systems like this, we found out that this protein isn't really just a target. We found that actually the electrostatic surface potential, or the surface there is interlink kinase, the cartoon is actually an inhibitor molecule. What we found is that we don't actually need to upregulate the protein. What we need to disrupt the interaction between those 2 proteins. We mimicked human physiology, we did some drug discovery, and now we're able to move that forward. And I think without proper systems that recapitulate human physiology, we're not going to be able to do that. So obviously one big problem in heart failure is the cost of trials, right? So I'm going to give you an example of one strategy we're taking in our intellectual property office to mitigate the cost of large trials. And that's using disease indications as a gateway. So everybody in this room knows that a heart failure trial is very expensive, and you often need thousands or tens of thousands of patients to show a difference. So let me show you an example of where you don't need to do that and how you can potentially get around that. So inhibition of HMG-CoA reductase, very familiar to people who do cardiac research, was initially approved and statins developed specifically for familial hypercholesterolemia, all right? Initial approval was for FH, and went through clinical trials, an orphan disease indication, and an FDA voucher. At that time, after a smaller, more focused trial that allowed for quicker integration into the market, now we could expand to, or they could expand into broader dyslipidemia indications. So this is another way in which you can focus small regions of potential cardiac dysfunction, get your foot in the market, rather than just saying, I'm going to take on a large population of heart failure. Because this is, every time we go to a venture capital group, every time we go to a pharma partner, heart failure is not high on their list because of the cost of the trials. So just in conclusion, I talked about the translations of targets, molecules, or pathways. I'm not just talking about specific proteins or RNA or so forth. I'm talking about even pathways. Suffers, and I'll use the word suffer, from great hypotheses. So I think earlier evaluation and adjudication of those pathways is needed. And translation doesn't come easy to preclinical or clinical scientists. We're not trained for it necessarily. So you need to establish better translational teams, and that's what we've done at SickKids. We've even brought in an entrepreneur in residence to try to do this. And I think earlier integration of multiple disciplines, and I don't just mean academic, non-academic, and intellectual property groups, using clinical data and samples to tie back to the last set of talks. And I think we need new clinical funding strategies that allow this to happen. And I don't just mean funding IP&C work, but even funding cross-disciplinary teams with high-risk, high-reward projects that allow you to investigate these diseases and move things forward in a traditional way that's not covered by normal funding agencies. Thank you very much.